This slide recapitulates what we have talked about in the first podcast on the GI system. The mucosa with an epithelium, a lamina propria, and a muscularis mucosae, as shown here. The submucosa, a connective tissue layer, here showing some submucosal glands. You may also find lymph nodes in the submucosa. We'll see those nicely in the laboratory. The muscularis externa with an inner circular, outer longitudinal layer of smooth muscle, and then a serosal or adventitial layer. So this reflects the general overview or plan for the GI system. Recapitulating one more time, the mucosa with an epithelium, a lamina propria, muscularis mucosae. As we've said, the epithelium will vary depending on the organ. It may be glandular depending on the organ. The lamina propria will be rich in connective tissue with cells, capillaries, lymphatics. The muscularis mucosae is always smooth muscle. There's an inner circular and an outer longitudinal layer of smooth muscle. The muscularis mucosae, while always present, isn't always apparent on all the histological sections. The muscularis mucosae essentially provides some structural support for the epithelium, and its contractions will help to empty some of the mucosal glands. This is critically important as we get into, for example, the small intestine or the large intestine. Now, the submucosa, as we've suggested, is a connective tissue layer. There are blood vessels, lymphatics, parasympathetic ganglion. There may be glands present in the submucosa depending upon the organ. The muscularis externa is generally smooth muscle, the exception being that in the upper region of the esophagus you see skeletal muscle, and in the middle region of the esophagus you see intermingled skeletal and smooth muscle, and this may be the only place in the body that I can think of where there's intermingling of skeletal and smooth muscle. The muscularis externa consists of an inner circular and an outer longitudinal layer of smooth muscle generally. Sometimes the stomach picks up a third oblique inner layer. There are oftentimes a parasympathetic ganglion between the layers of the muscularis externa. As we've said, the serosa with a mesothelium or the adventitia if the organ is buried within connective tissue of other parts of the body. Lymphoid tissues, we've talked about mucosal associated lymphatic tissue and gut associated lymphatic tissue. Oftentimes, lymphatic nodules in the GI tract will disrupt the muscularis mucosae. Now, you have to be a little bit careful because sometimes there will be associated lymph nodes that you'll see in histological sections. These will be distinct organs that will be outside the serosal layer of the GI tract. So let's spend a few minutes and talk about the esophagus. The esophagus functions to transport food to the stomach. It's lined by a stratified squamous epithelium. It's lubricated by mucus glands. These mucus glands can be mucosal or submucosal glands. The gland ducts open to simple columnar epithelium on the esophagus, and this is kind of clinically important because where the gland ducts open onto the esophagus, that's an area that can be prone to hyperplasia and to cancer. With respect to the muscularis externa of the esophagus, the upper third of the organ has skeletal muscle. The middle third is skeletal and smooth muscle. The lower third of the esophagus is uniquely smooth muscle. This is just a low-power histological view of the esophagus. So here is the lumen, the mucosa shown like so. You see the epithelium. It's difficult at this low-power magnification to pick out much of the lamina propria or the muscularis mucosae. Here is the submucosa. You can notice some lymphatic nodules in the esophagus. This nodule is probably disrupting the muscularis mucosae and might actually be in the lamina propria. Here's a mucus gland in the submucosa. Oftentimes you see mucus glands in the esophagus. And here is part of the muscularis externa and a little bit of the adventitia on the organ. A little bit higher power view. On the left side is a much higher higher power view of the mucosa, so you can see nicely the epithelium, the lamina propria, and a portion of the muscularis mucosae. The image on the right is a little bit lower power view, but it still shows the mucosa nicely with the defined muscularis mucosae. Look at the large submucosal gland in the lamina propria. Here's a duct for that gland, and then showing a portion of the muscularis externa. Another histological view of the esophagus to make the point. A stratified squamous epithelium. This is a high power view of the mucosa, so a stratified squamous epithelium. Look at this lymphatic nodule within the lamina propria. Here it's actually disrupted the muscularis mucosae. You can see a lot of diffuse lymphoid tissue around this nodule. You can even see some of this lymphoid tissue invading the epithelium. Here we can see on the little bit lower power view the submucosa like this, and then the muscularis externa. You begin to see the inner circular and outer longitudinal layers of the muscularis externa, and then a little bit of the adventitia to the bottom part of this image. If we talk about the stomach and the intestines, a couple of points I'd like to make. The surface area in the stomach and the small intestine is increased by gross folds. In the stomach, these folds are called the rugae, and in the small intestine, these are called plica circularis. Now I'm going to drop out the bottom part of this slide. We'll pick it up in a later podcast. Basically, the rugae and the plica will increase the surface area over a simple tube, and in the stomach there's an increase in surface area by something like 3 times as if it was just a simple tube. And these rugae in the stomach and the plica in the small intestine consist of the mucosa and the submucosa. With respect to the stomach functions to store and digest food. Almost a liter of acid can be secreted in the stomach. That acid has an enzyme mix, and it's secreted by the epithelial lining, and this occurs essentially every time you take a meal. You'll secrete about a liter of fluid into the stomach. The mucosa shows an abrupt change to a simple columnar epithelium. So you've got the stratified squamous epithelium of the esophagus transitioning very abruptly to the simple columnar epithelium in the stomach. The mucosa of the stomach is very highly glandular, and depending on the region of the stomach, there are very distinct cell types that can be identified in the mucosa of the stomach. Regional differences in the stomach tend to be based on the mucosal lining. The muscularis externa of the stomach is very thick, and in the fundus or the body of the stomach you actually pick up a third internal oblique layer of smooth muscle. I like this image because it shows the very abrupt transition from the stratified squamous epithelium of the esophagus to the simple columnar epithelium of the stomach. It's literally like the two organs are stuck together. There's a very abrupt transition. This cartoon just shows the general arrangement of the mucosa in the stomach. It's folded, and you'll see pits and you'll see glands. So this would be a pit, an invagination, a little bit of the lumen. The lumen of the organ would be up here. So the pit is an invagination of that lumen. Then you have the glands, which are further invaginations of the epithelial surface. And so I could put my pointer here to the lumen of the gland and trace that all the way up, like so, coming into the pit, and that area is continuous with the lumen of the organ, and as I said, that area is actually essentially space outside of the body. Here's a cross-section of the gland like so, at the base of the gland, so this would be the lumen of the gland. Here would be lamina propria and the muscularis mucosae shown on the bottom. So this just is an overall view of the mucosa in the stomach. Now there are some regional differences in the stomach that we can define, and would like you to at least be generally aware of these from the point of view of histology. We can talk about the cardiac region of the stomach, and the cardiac region of the stomach has relatively short pits and short glands, but the glands are of equal length. And the epithelium consists mostly of mucus cells. The fundus of the stomach has relatively short pits and very long or deep glands. There are multiple cell types that can be identified in the fundus of the stomach. We'll talk about those in a few minutes. And then the pylorus of the stomach has very long pits and relatively short glands. Once again, the epithelium in the pylorus of the stomach is mostly mucus cells. Now, on its face, it is sometimes difficult to distinguish the cardiac and pyloric regions of the stomach just by looking at the surface epithelium, and it is sometimes a little bit tricky to be able to tell if the pits are long or short in relation to the glands. So, for our course, we're going to expect you to be able to identify the fundic part of the stomach because, one, it's going to be easy to identify the long glands and the multiple cell types in the fundus of the stomach. If we had the cardiac portion of the stomach on a slide, we would only expect you to identify it as the cardiac stomach if there was a profile of the esophagus on the slide. In the same token, if we had the pyloric portion of the stomach on a slide, would only ask you to identify it as the pylorus if we had a section of the duodenum on the slide. And this will become clear to you in the laboratory. Here is a histological view of the mucosa of the fundus of the stomach, showing a nice diagram on the right, and it's showing you the pit, like so. So the pit would be like this, and then the gland itself, the deep gland, and notice that it branches a little bit. Look at the varieties of cell types in the gland. And we make this point about the isthmus because the isthmus is right at the boundary between the pit and the start of the gland. This is where the stem cells in the stomach are located. So cells that are actively dividing in the stomach to give rise to the mucosal lining are actually found in the isthmus. Here we can see a variety of cell types. You can see the mucosal folds, the muscularis mucosae, and here a little bit of the submucosa. This would show a profile of one of these pits and the gland coming into the pit with a variety of different cells. So we'll talk about the surface lining cells, the stem cells or regenerative cells, mucous neck cells, parietal cells also called oxynitic cells, the zymogenic cells also called chief cells, and the enteroendocrine cell or APUD cell. The diagram is a little bit misleading here because it's showing these enteroendocrine cells and it's showing their surfaces touching the lumen. That is not always the case. Some of the APUD cells do indeed touch the lumen, but many of them do not touch the lumen, and we'll talk about that in just a few minutes. So various cell types that are in the fundus or the body of the stomach. The surface lining cell, shown diagrammatically like this, is a mucus secreting cell. It's a tall columnar cell. The apices tend to be relatively clear. It secretes a thick neutral to alkaline mucus, and this offers mechanical protection to the epithelium in the stomach. Also will have some antibacterial substances in that thick mucus. The mucous neck cell is throughout the gland, but it's mostly in the neck of the gland. It secretes a thin to acidic mucus. This mucus will mix with and lubricate the chyme. The stem cell or regenerative cell are the mitotically active cells. These are found in the neck region or near the isthmus of the gland. The parietal cell, also called an oxynitic cell, is throughout the gland, but they're mostly in the upper half of the gland. These cells have what I like to call a fried egg appearance. They're a large eosinophilic cell when they're stained with H&E, they have abundant folds that you can see at the electron microscopic level. These folds are on the apical and basal lateral surfaces. These cells have abundant mitochondria. They have canoniculi with microvilli to increase surface area. These cells are the cells that secrete HCl. They also secrete gastric intrinsic factor, which functions to bind vitamin B12. There's the chief cell or zymogenic cell. These cells are located throughout the gland, but they're mostly in the lower half of the gland. These are polarized for protein secretion, a typical protein secretory cell. These are the cells in the stomach that secrete pepsinogen and renin. And finally, the enteroendocrine cells, also called APUD cells or DNES cells. There are many cell types like this. They're located throughout the epithelium, in the glands and on the surface in the stomach. They're also in the small and large intestine, and they are in other organs in the body. These cells secrete many important products. APUD, I won't ask you to remember this, but it stands for amine production, uptake, and decarboxylation. DNES stands for diffuse neuroendocrine cells. These are all basically defining the same type of cell. These cells secrete essentially hormone products that play important roles in various functions of the GI tract. And it is notable some of the APUD cells are actually derived from the neural crest. The top image here just shows some of the GI hormones, and you'll hear about these in physiology. We'll come back to this in a later podcast. This just shows where these hormones are secreted, whether you're talking about the fundus of the stomach or the duodenum or the colon, for example. The diagram on the left is an enteroendocrine cell, and here it's showing this enteroendocrine cell, and it's showing that this cell is actually polarized for secretion, but the secretion is towards the basal lamina, because these are products that are secreted into the bloodstream to act either on an endocrine manner to distant areas in the body, or these could act in a paracrine manner, secreted into the lamina propria, where they'll interact with other epithelial cells in the GI tract. So this is an enteroendocrine cell, and here on either side of it would be epithelial cells that are surrounding it. So the lumen of the GI tract would be here. Now, this cell would be a cell that we call a closed cell. There are some cells that are called open cells. They are also thought to be enteroendocrine cells, but their apical boundary does reach the lumen of the organ, and these cells may function to regulate hormone secretion by the other endocrine cells, and it's thought they may actually have taste-like receptors that are on these surfaces. Secretion of the APUD cells is regulated in a paracrine manner, and it's regulated in a paracrine manner presumably from secretions of these open cells and maybe also from neural inputs. Here's a nice profile of the mucosa of the stomach. Here, the fundus of the stomach. Notice the shallow pits and the relatively long glands. Lamina propria like so, muscularis mucosae like so, and a little bit on the bottom of the slide of the submucosa. A higher power view of the epithelium in the fundus of the stomach. Look at the nice distinction between the parietal cells and chief cells. Notice the fried egg appearance of these parietal cells. Very, very eosinophilic in nature. As I said, the parietal cells tend to be located more towards the top portions of the glands, but look, there are some that go very deep in the gland. And then the chief cells tend to be located more in the base of the gland, but look, there are some that go up a little bit higher in the gland. Here, just a diagrammatic view of the parietal cell and the chief cell. As we said, the parietal cell produces HCl and gastric intrinsic factor. Notice the large intracellular canoniculi, large infoldings of these membranes. lots of mitochondria here. These cells secrete HCl, and they secrete these against a concentration gradient, so there's a lot of active transport for these cells to secrete HCl into the lumen. The zymogen secreting chief cells, a typical protein secreting cell, basal rough ER, Golgi, and apical zymogen granules. These zymogen granules are held for release when the cells receive signals to release them. Here's just an electron micrograph of the parietal cell. Notice the large intracellular cananiculi and the abundant mitochondria associated with them. And then a chief cell, the basal nucleus rough ER, and notice the zymogen granules. And actually these are profiles of several different chief cells, probably 3 on the field, maybe like 1 here, 1 here, 1 here, the nucleus only in the plane of section here. And again, these zymogen granules are held until cells receive a signal for secretion.